Ladies and gentlemen, Dr. Andrew Kaufman.
Thank you. Thank you, Don. All right, as my mic is working, great. Good morning, everyone. I'll tell you, it's fantastic to be in a room full of people close together who are not dressed up like surgeons. So I want to apologize in advance because I'm going to be talking a little bit about science and math today, and I know that's not very fun for people, but it's really, really important in this situation because our ignorance of the science and math and our fear of looking at it and our difficulty with the language that it's expressed in is allowing us to really be taken advantage of. And I feel like it's really my mission to try and expose this as much as I can, along with some other brave people who have been great contributors. So we're going to do that a little bit today, but I'm going to try to make this as relevant as possible so that you could see how it could really affect you in the real world. And we're going to be really talking about the entire history, in a sense, of this pseudoscientific, dogmatic field of virology. So why don't we just dive right in. I found this image when I was just searching for images related to fraud. I thought it was hilarious because here the government is warning us about people scamming us based on the coronavirus when the whole darn thing is a scam. So we're going to start off talking about this SARS-CoV-2 virus. It's not really a virus, but they call it a virus that is allegedly behind this pandemic. And then we're going to go backwards in time from there. So what did they actually do to prove that this virus exists? Because that's the main proposition that leads to all of the downstream effects, including all of the policies we've been subjected to. And what they did is basically they took some lung fluid, right, pus from the lungs of sick people, by putting a tube down in there and swishing around some salt water. And from that messy sample, which contains all kinds of junk, right, it's basically rotting flesh from a sick individual, they took out some small fragments of genetic material. And from that, and it seemed like this was their main priority, is to develop a PCR test. So just so you know that they found these small little fragments of genetic material not just by looking at everything that's there, but they had to look for something specific in order to use this PCR technique to find a needle in a haystack. And what they did was they just took sequences from another virus, SARS-CoV-1, which they also used the same exact techniques to discover, and they said, oh, look, we found similar sequences of short genetic strands, not an entire gene, not an entire genome of an organism. Oh, there we go. But little tiny fragments of just 200 to 300 bases long, not enough to do anything, and it's unknown what the significance of those are in reality. They also claim to isolate the virus, but based on this historical procedure I'm going to discuss today, isolate has a totally different meaning if you're a virologist. And what it means actually is to mix some rotting flesh from a sick person with a cell culture that first you starve and then you poison. And you put those things together and then you wait to see damage or disease or death of those cells in the culture, and then they call that a virus. Now, there's one big thing that they're missing in these experiments, which is a control. And I'm going to talk more about that in a moment. But here is the method that is really the way that you can isolate a virus. And this has been done many times with bacteriophages, which are what most people think of as a virus of bacterial cells, and with other phage-like particles that are associated with algae or amoeba. By the way, they've also been able to use this exact procedure to isolate exosomes, which are little extracellular vesicles or little sacs of fluid or particles that look just like what they say virus particles look like. You see them with damaged and dying cells, and they can purify those out by this technique that's never been used for an alleged pathogenic virus. And what they do is they take the same sample of rotting tissue from the sick person, as you can see, but in this case, they actually purify it. So they filter it, which removes all the big bacterial cells. It can't remove everything because some bacteria may exist as spores or that pass through the filter. But it's a good step. It removes all the human cells for sure. And then they use a technique called density gradient centrifugation. And this is not really fancy, even though it has that fancy name. It's been around for a really, really long time. And they're just spinning around the fluid in a test tube with a special medium in there that allows things to separate by density. So in the test tube, you have this little band, and all the things in that band have the same physical property of density. And you can basically separate the little particles in that band, and then you can take it out, and it's pure. And once you have a pure sample, then you actually have an intact thing, right? You can look at it under the microscope and see all these things. They all look the same. And then you can analyze them chemically and structurally and see exactly what they're made of. And from there, you can do further research. So pretty straightforward. What they call isolation for viruses that are supposedly cause disease in humans is totally different. It's this recipe that I described where they take out short sequences of genetic material of unknown significance. They match those to a library, a computer library of other sequences that they got the same way that they also say are viruses. And then what they do is they mix it with that starving cell culture with toxic chemicals in it like antibiotics and calf serum, like part of the blood from fetal cows. And then they let that grow for a period of time until they see the damage. Now, if they were to do this experiment correctly, they would have to also do the same exact thing, but use the lung fluid from healthy people as well, right? Because how do we know that what's causing the cell damage is from the lung fluid, or is it from the constituents of the cell culture? So that's the control experiment that I'm talking about. It's never been done in the history of these experiments. Now, these are the cytopathic effects, and this is the only slide with microscope images, so I apologize, but I just wanted to show these because you can see all these different images in here from different viruses, and you can see that they all have similar characteristics. So what they're doing in each of these things is they're taking some people or even one person with a specific illness, right? A herpes outbreak, a cold, et cetera. And then they are mixing that with the same recipe of cell culture that's starved with poison. And they're seeing these cytopathic effects, they call them, which is this disease cell damage. Now also, when they show images of the virus that are not computer graphics, they're almost always pictures of this where they zoom in and they show particles either inside of a cell or coming off of a cell. And they say those particles are the virus, but how do you know? Because we do know there's lots of different particles that come off dying cells. Okay, so let's shift and talk a little bit about the PCR test, a test that is really meaningless. But just to make sure we review the important points, so it tests for short genetic sequences that we don't really know where they came from. There's no gold standard for this diagnostic test. So when I was in medical school, I was taught it's very important when you have a new test, there has to be a gold standard to compare it to, so you can know what the accuracy is or the error rate. Now in this situation, since a virus has never been isolated or demonstrated to exist, and by the way, if you look in Off Guardian, there's an article by Torsten Engelbrecht called Why the PCR Test is Scientifically Meaningless. And in that article, he actually corresponded with authors of several of the major papers claiming to isolate a virus and asked if basically any of those labs had purified the virus or had any samples where there was just virus. And all of them replied that they had not done this. So they admitted it in writing, and that's published. So that's a really important resource. So there's never been tested against a gold standard of showing a virus, so there's no way to know if it's accurate or not. And in fact, there was only one study that was published that tried to estimate the accuracy of the test, and it estimated the false positive rate to be 80%, which basically makes the test something that should go right in the trash. Okay? Now, other entities have estimated this false positive rate, but it cannot be calculated because there's nothing to compare it to. So one of the consequences of this testing, and we've all seen case numbers being reported to go up in various places, and that is the main policy driving statistic. And when they say case numbers, they're not talking about people who are actually sick. They're talking about people with positive tests. So that's why this is so important. Now, Operation Moonshot is a plan in the United Kingdom that's been put forth by the government there, and it's been described in the British Medical Journal. You can see the reference at the bottom. And what this plan is about setting up a testing industrial complex, essentially, that is going to ramp up, and by 2020 is going to allow weekly testing of the entire British population. So let's see what the significance of that might mean for the people of the United Kingdom. Okay, so full rollout is what they call it. They're targeting for the beginning of 2021, so only a few months away. And they want to have 10 million tests every day and the entire population tested each week. Now, the British government has put out some numbers about the prevalence of COVID-19 infections and about the estimated false positive rate. And they get that from other PCR tests used in other viruses, which also don't have solid data because there's no gold standard in those tests either. But they gave that a false positive rate of 2.3%. So we'll just use that. We'll go with it since it's put out by their own government. So the prevalence that they said the most recent week in September, where they had data, was 1 in 500 or 0.2%. Okay, so if we apply this to the entire population, or to 10 million people that we're going to test, we'll see that a 2.3% false positive rate, and what a false positive is, it's the number of people who are not sick, who don't have any disease, will give a positive test. So we know that if 0.2% prevalence, that's who are sick, out of 10 million, that's 20,000 people. But the false positives will be 230,000. Okay, so many, many times the amount of actual cases. So they could put out a headline, which would be very shocking and worrying, that there are 250,000 new cases in one day from testing 10 million people. However, only 20,000 out of those 250,000 will actually be positive. Now, if we apply this to the whole week, the entire population, you'll see that 1.56 million people each week just in the United Kingdom will be labeled as false positive. And this policy has consequences based on the testing results. So you're going to be required to have a digital passport, which is going to say what your weekly testing results are. And if they were positive, you're not going to have access to work. You're not going to be able to go to in-person healthcare appointments. You will not be able to attend venues such as the present one. And you won't be able to fly, and they specifically mention flying because that's international travel they're talking about. So 1.5+ million people in the UK each week will be falsely prevented from engaging in these activities based on a faulty test. This is a real, real problem. It's coming soon, and something needs to be done. Let's get into another layer of fraud, and this fraud is about causation, so Koch's postulates. Now, my critics have said that Koch's postulates is not relevant to viruses, but I ask you then, why do they keep publishing papers saying that they've proven Koch's postulates for viruses, including for COVID-19 related illness? So just to review what Koch's postulates are, and these were formulated a long time ago to be the rules of how you prove that a germ causes a disease in the germ theory. Robert Koch was a German physician, and their CDC is named after him, called the Robert Koch Institute. So the first postulate, the microorganism must be found in people who are sick, but not in people who are healthy. The second one, you should be able to isolate and purify the organism from the diseased person. Now, growing it in a pure culture has to do with bacteria. You can't do that with a virus if a virus exists. And then when you put that cultured organism or that purified sample of particles into a healthy host, usually animals are used for these experiments, it should cause the same disease, okay? Not just make them sick, but cause the same disease. And then you should be able to re-isolate it from that person. Okay, pretty straightforward. These are based in logic. They make sense. So here is one of the recent articles where they claim to satisfy Koch's postulates for the SARS-CoV-2. And what they do in this case is they take a sample from another study, which is not a purified virus, okay? So right there it fails because they didn't do the first step, but I'll give them a pass for now. They also put this into genetically modified hosts, not natural hosts. Okay, so that could affect it. All right, but I'll give them a pass on that too. And here's what they said. Our results, blah, 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 completely satisfies Koch's postulates. They said in combination with another paper. Okay, so I'll give them a pass on that first part, but we'll focus on what they did in this paper. And when they say Koch's postulates, they reference Rivers, okay? This famous article from 1937, written by Thomas Rivers, who was one of the major figures in virology. And he wrote, it's obvious that Koch's postulates have not been satisfied in viral diseases. And essentially this paper was trying to say, if we lighten up on some of these things, maybe we can prove viruses cause disease. Okay, so we're going to go with that. And Rivers said he relied on these two quotations from Robert Koch to modify the criteria. And you can just, I'm going to skip by a little bit to make sure we finish on time. But essentially it boils down to these four aspects, very similar to what I already stated. We're going to focus on the fourth one in this paper to show you the fraud. And basically that is that the isolated parasite or virus produces the same disease with all of its characteristics in a normal host, and we'll give them a pass on the normal host. So, right here, they reported the clinical manifestations, which is like signs and symptoms of what we can observe about these animals after they were given this so-called virus. Now, we had three groups, right? The experimental group, that were the genetically modified mice given the actual virus. You have the second group, we have regular mice given the virus. And the third group, we have genetically modified mice given phosphate buffered solution, which is just like water with electrolytes. Okay? Now, look at how many of them had symptoms in these three groups. You see it's low numbers, and it's less than half in all the groups. So I did a little statistical analysis to show if these groups are actually different from each other or the same. In other words, maybe they just all showed some symptoms, and it doesn't matter what you put up their nose. Okay? And that's what the statistic shows. These groups are not different from each other. So right there, the experiment fails, but they didn't do that analysis in the paper. They just focused on the 37% and said it was significant. So let's look at what these clinical manifestations were. And I want to poll the audience. If you could just shout out, what are the symptoms of COVID-19? Well, what are the real ones that are reproduced everywhere. I know that they keep adding more, right? What? Fever. Cough. Shortness of breath. Good. Fever, cough, and difficulty breathing, right? I mean, I know that a lot of symptoms have been attributed to this. We can't ask mice if they can smell things. But the respiratory symptoms are the main thing, right? This causes a type of pneumonia. Right? It causes respiratory distress syndrome, right? So there's got to be pulmonary symptoms, so shortness of breath, cough, sputum, like coughing up stuff, sputum production, something like this, right? And then fever. We all know fever, right? Those are the most common things. Okay, they're taking temperatures of people everywhere for this, right? So we'd expect at least fever. So what symptoms did they actually have? Weight loss and slightly bristled fur. Anyone hear news reports of bristled fur in COVID patients? Weight loss also not a symptom of COVID-19 that's reported. It's a very non-specific symptom. So once again, the experiment fails on this count that this is not the same symptoms as the disease in question. Now we go to the autopsy findings. Now, there's really a lack of data here because normally in a health crisis situation, they're supposed to autopsy everyone who dies suspiciously. They specifically reversed this policy worldwide during this pandemic. And I believe to cover up the true cause of death. Okay, but they would say that maybe it was too dangerous to spread the infection or something silly. But you know, because pathologists, they can take precautions. People work with these things in the laboratory. That's how they develop the tests. So they should be able to work safely with the bodies. But nonetheless, so we do have 2 studies that were published that do describe autopsy findings in a case series of people with this sort of pneumonia illness, right, that they're calling COVID-19. And what they found is the same thing, basically. So you see similar characteristics. Diffuse alveolar damage just means to the air sacs where the gas is exchanged. There's damage. There's hyaline membranes, which you can see under a microscope. There's metaplasia, which is a little bit distinctive, and that's like kind of precancerous cells or cells that develop abnormally. And then blood clots. And that was a very characteristic finding, blood clots in all the autopsy samples. So what do we find in the autopsies of the mice? Well, we found interstitial pneumonia with macrophage infiltration. There was no macrophage, that's a type of white blood cell. They weren't found in the human studies. No hyaline membranes were found. No edema, no metaplasia, and no blood clots. So essentially, the autopsy findings didn't match. Once again, this article fails. So what are my conclusions? None of the postulates were satisfied. And then despite taking a toxic liquid from monkey cells grown with infected lung pus and poisonous chemicals being inserted into the mice noses, less than half showed any symptoms at all. And none of them showed symptoms or autopsy findings consistent with COVID-19. So a big fail, fraudulent paper. And I'll tell you that paper even has more fraud than that in it, because here's the next level of fraud I'm going to talk about, which is fraudulent claims that have been made in scientific papers. So right back to that paper, in Nature, one of the most prestigious journals in science. And right in the first sentence of that paper, in the introduction, it says, right, that COVID-19, which is caused by SARS-CoV-2. That is a declarative statement, right? It's definitive. It says that this illness is caused by this virus, and that it must imply that there's solid evidence behind that, right? And certainly, if you go down any street in the world right now and ask them what's making people sick, they're going to say this virus, right? There aren't many people like us who know the truth about this. So they listed 2 references there at the bottom, 1 and 3. So I went and looked. Here's number 1. I was very familiar with this article from the New England Journal, where it says that the virus is likely to have been the cause. and that their study doesn't fulfill Koch's postulates, once again mentioned in the virus literature, showing its relevance. It only has evidence implicating it in an outbreak. Implicating, likely to have been, very different. Okay? So this paper didn't give the evidence for that statement. What about reference 3? This is from the Chinese Medical Journal, and in that article it says associated. Right? Now we all know association or correlation does not prove causation. Okay? And you know for sure these scientists who have PhDs also know that, yet they still made this fraudulent claim. I apologize. Oh, that you can read it, actually. Okay, so this is from another paper, the Korean paper claiming to isolate the virus. And they said also it's identified as the causative agent. Once again, a declarative statement. This is in the introduction section of their paper, not the conclusions. They give a reference here, number 7, okay, which is actually a genome study. So I looked at number 7. In there, it actually doesn't do any experiments in number 7 that could show causation. They're just doing genetic sequencing. So they just have in the intro that there's an association, and their conclusion that the genetic features have a potential association. So not even strong enough for a full association, only a potential one. And they're using this to claim proof of causation. Okay, this was perpetuated forward on the McMaster University website, where they also claim to have isolated this virus. And I'm waiting for their article to be published so I could see their methods. But I imagine it's the same as everyone else's. And they said it's set in motion the pandemic. So that's a pretty controversial or statement. So essentially, association implication gets changed into is the causative agent, set in motion the pandemic. And this is what, of course, people believe, because these are very strong statements. And why would you think that they're actually not backed up by any scientific evidence? So let's get into another area of the fraud, the genome, which I just alluded to. Now, this is a very striking image of basically two puzzles, and I guess what we can learn is that the puzzle manufacturers have one template for their jigsaw pieces, but also this is kind of like how the genome of viruses are put together and said to be real, even though they're very theoretical, okay, because they're just taking little pieces from different things and they're patching them together, right? And you can see this is neither a horse nor a train because it's a mixture, okay? We have another one here. I could go all day with these. I really like these images. If anyone wants to send me some puzzles, no. So this is the technique that is in every paper I've seen on the genome. They call it next generation sequencing. Right? Not to be confused with Star Trek: The Next Generation. But this is a very powerful technique because if you were to take an entire genome, like let's say, you know, the human one where we had a lot of chromosomes and really long strands, and do a whole chromosome, you know, start at one end and go all the way to the other end, it would take weeks. Right? That's called the Sanger method. And that is still done, by the way, and it can be combined with this. But this is a way to take a super long strand and cut it into small bits, but you know exactly where you're cutting, and then take all those smaller bits, which is called the cDNA library, and you put them on a matrix, right? Not the matrix, but a matrix, like a paper, and they attach at one end, and they all kind of stick up like little strings coming off the paper, and then you can sequence all of those at once, at one time. So it takes weeks of work into hours of work. Okay, so very, very powerful. But in order to use this properly, and what it's really intended to be used for, is to find genetic mutations in an organism that you already know the genome sequence. It's not the technique to determine the genome sequence of a brand new, yet undiscovered organism. Okay? And the reason is because what we have here, when we take the lung fluid from a sick person with pneumonia, and we only have short fragments. So we don't start with one intact genome and then chop it down so that we can sequence it faster. We start with, in one paper, it was over 20,000 small fragments. Okay, the fragments are about 200 to 300 letters long, and they say the genome is 30,000 letters long. So you can see it's going to take a lot of pieces to make that up. Okay, but since these things, so they don't start with an intact organism, right? Like let's say you take a mouse, and then you take out some mouse cells, and the mouse cells are just mouse cells, and then from those mouse cells, you extract the nucleus and the DNA in the nucleus. Then you can chop it up and sequence it because you know that it came from mouse cells. In these experiments, there's no intact virus particle that they pull it out of and then chop it up. They take it out of this lung fluid where they can't find any viral particles in the lung fluid directly. They haven't demonstrated that. It just has a mixture of genetic material from our own cells, our bacterial cells, fungal cells, anything that could be in our lungs at the time that we're sick. So how do they know where these sequences come from? And what they do is after they sequence the 20,000 little strands, then they put them in a computer program. Okay, and this is called sequence alignment. And this computer program essentially builds the puzzle. And it puts all these little fragments together. Now here's the problem. Sometimes they might have an overlapping complementary sequence, and you say, okay, these fit together like that. But many times they don't. So there are gaps. So what do they do with the gaps in the sequence? Well, they make it up. They pull it from that old computer library with all the other viral sequences that were made the same way, and they pop out one that looks like it might be right. Maybe it has some homology on both ends of where the hole is, and they stick it in there. And of course, this is done by a computer, right? Because if we did this by hand, it would take probably 6 to 12 months. Okay, but a computer can do this quite quickly. So you end up with this big, long length. We don't know what the length really should be, right? Because it's never been taken out of a pure organism. And they just hack it together on the computer, and then they say this is the viral genome. And they even go further to lie about this and say that when different labs do this in different parts of the world, the sequences don't match up exactly, and even the length doesn't match up. Like imagine if some of us humans had one extra chromosome, you know? And they say it's because of mutations of the virus. Of course, that's their explanation, but really they're different because they're not anything at all in the first place. They're just making something from scratch. So of course, if I decide to go on the pottery wheel and make a bowl, it's going to look a little bit different from when you make a bowl on the pottery wheel, right? Even if we're both highly skilled. So that's all you're seeing there. So once again, this genetic sequence fraud. Okay, now I'm going to go back in time a little bit and give you some historical context. Okay? We're going to go back to, originally there was this theory of miasma, that there was some noxious substance in the air that caused disease and could spread. Okay? And there was never real proof of that, but that stuck around for a while. When they started using microscopes after they were invented, they saw microorganisms that before were unknown. Okay? And when they looked at tissue from sick people or animals under the microscope, they saw these microorganisms like bacteria and fungi. They made an assumption that those microorganisms were causing the disease, not considering that they may merely be present and have another function. Okay, so once again, this error about association or correlation and causation. Okay, it's one of the biggest logic errors that's made in science, and whenever it comes up, it's a real big one you got to pay attention to. So they were able to see bacteria in some diseases and said that that was the explanation of the cause of those diseases, but there were others that remained unable to find an organism. And poliomyelitis was one of those. So what they did was they theorized that there was actually a poison, a toxin, that came from somewhere, and this was what a virus was. And that's why the Latin root of virus means poison. And they thought that this was a transmissible type of poison that could actually reproduce in your body, though. And this is what makes it a very unique and special type of poison that deserves a special name of virus. Okay, and so what they would do is they would have a sick animal, and they would filter out the diseased part of their tissue, like I described in that purification experiment, to remove cells, cellular debris, but it doesn't remove everything. And then they would take that filtered material, okay, and they could make other cells sick with it. Okay, so this was what they thought a virus was. However, they were really not able to see it ever. And when the electron microscope came out around 1930, they really were hopeful, but it never panned out. They could never see this kind of protein poison virus that they were theorizing. Okay, so essentially the field was not very successful, and it was sort of collapsing in on itself. Okay, but then a couple of things happened to revive it. Okay, and this man, John Enders, is a very key figure in this revival that led to this modern dogma of virology that I described with the current situation. So one factor was that in the early 1950s, the discovery of the double helix of DNA was made public. And this was like a new beginning in biology, that the importance of this genetic molecule was going to transform our understanding of everything, and including viruses. So this is a very key occurrence. And has to do with what was going on with vaccines. And Enders was very successful in, he is actually the one that came up with the scientific idea behind Salk's poliovirus. But then he kind of bowed out and let other scientists develop the idea and get the credit for it. But
hi, this is Chris Olson from Treasure Island Coins coming to you.
Just a brief message from our sponsor. So anyway, the Salk polio vaccine did not go well, and Enders was pretty critical, and he decided that he wanted to focus on measles going forward. So at the time, the virologists who were studying this, they thought that taking that filtered material that they called a virus, that it would only affect the same exact type of tissue that the person who was sick had. So, for example, if it caused pneumonia, then it would only affect lung tissue, and that it wouldn't cause any damage to other types of tissue. But Enders had this idea that maybe it could affect other tissues, and maybe, and with the polio vaccine, it was actually used, it was cultured in fetal tissue. And then he had this idea that maybe it could be in other animal cell cultures. So this study in 1954 was critical, and it basically laid out the modern experiment to prove a virus. But it does something more than that I'll get to in a few minutes. So what I discussed in the beginning about mixing the rotting flesh with starving, poisoned cell culture that comes from this paper. And what he did was he found some kids with measles, and he took nasal swabs and washings and such, and put that in some various cultures, and he used monkey kidney cells. Okay, this is the first time these monkey kidney cells, and if any of you have heard my lectures, I mention monkey kidney cells quite a bit, because they're the favorite cells used in these experiments. And so he was able to, what they call this, propagate, because he would put the infected tissue in with one culture, let it incubate until it showed damage, then take liquid from that, filter it, and put it in a second one, and that showed damage as well. Okay? And so that was the main accomplishment. And I brought this paper because I didn't have it at the time I did my slides, but I wanted to read what he said in his conclusion section about this, okay? Because he changed his tune after this. He wrote, "While there is no ground for concluding that the factors in vivo, that means in a living organism, are the same as those which underlie the formation of giant cells and nuclear disturbances in vitro," okay? So there's no ground for concluding that those factors is consistent with the properties that a priori might be associated with the virus of measles. Okay, let me translate that. They saw damage in the cells in the laboratory culture. He's saying there's no reason to think that's the same thing that what actually happens in the person with measles. Okay, so in other words, he's saying this experiment does not prove causation. He said there's no reason to conclude that it proves causation. Okay? So he was very critical of these techniques, and it was mostly a way to manufacture vaccines at the time. But what happened is he was awarded the Nobel Prize. He had lots of companies coming to him, giving him contracts, wanting his advice, etc. And he changed his tune about this line of critical analysis of his science and started to say that it was a cause. And this is really what I call the master virus dogma strategy here. So you take this recipe on the left, where you have rotting flesh from a sick person, mix it with poison in a starving cell culture for monkeys, and not only do you get the cytopathic effects that are quote-unquote proof of a virus without actually showing a virus, but also this is the recipe to make a vaccine. So that experiment I described before with the mice, what they essentially did was they put a vaccine in those mice without the adjuvants. Okay, because you take this cell culture product and add adjuvants, that's a live virus vaccine. Okay, if you take it out of that culture, put it in another culture, let that grow, take the fluid out, put it in another culture, and then put it in with the adjuvants, then that's an attenuated virus vaccine. Okay, so this strategy, which doesn't prove anything, according to the scientist himself, is now taken subsequently for every single pathologic virus, right? Every virus that causes disease. This exact technique is what is given as proof of the virus, and this exact technique is what's used to make a vaccine against that virus. And this is the deepest fraud behind everything else I mentioned that's been perpetrated upon us. So to summarize, we have many, many levels of fraud in this pandemic situation, in isolating viruses, in the diagnostic test, in showing causation of an illness, in sequencing the genome, and we have the underlying master strategy, right, where we can prove a virus and essentially make a new vaccine at the same time. Thank you, everyone, for listening.